el doctor Arturo Menchaca Rocha, presidente de la Academia Mexicana de Ciencias, el doctor Philip K. Maini, nuevo miembro correspondiente de la Academia, y el doctor Rafael Barrio Paredes, investigador de este instituto y anfitrión del doctor Maini. Escucharemos las palabras de bienvenida del doctor Manuel Torres, director del instituto.
Muy buenos días, doctor Arturo Menchaca, presidente de la Academia Mexicana de Ciencias, profesor Philip Maini, bienvenido a México, profesor Rafael Barrio, gracias por la organización de este evento. It is a pleasure to have all of you here for this ceremony in which Professor Philip Maini will be admitted as corresponding member of the Mexican Academy of Science. Professor Maini, a renowned and leading figure in the field of mathematical and computational modeling of biological phenomena, has had a long and fruitful collaboration with Mexican scientists. Professor Maini's extensive and refined use of mathematical tools to study biological systems has yielded significant results in the study of life science. The occasion to recognize the leading contribution of Professor Maini honors the Institute of Physics of this university. Our institute is further honored by the attendance of other distinguished professors to the event to follow. The third discussion meeting on patterning, segregation, differentiation in complex networks. I'm sure that you are all looking forward to the discussion of today and tomorrow, and I trust that many exciting ideas will emerge from this meeting. Professor Maini, I hope that you enjoy your visit to Mexico and our institutes, and there are many more to come. Congratulations.
Muchas gracias, doctor Torres. A continuación, toma la palabra el doctor Rafael Barrio Paredes.
It works. Well, thank you very much. I won't bore you telling you how I met Philip. I just want to say that I didn't know that mathematical biology existed before I met Philip. And his charm and his wise advice and his very juicy conversation made me change my field from physics to mathematical biology about 15 years ago. And I am very, very pleased to have met him and to collaborate him with along all these years. And I think this occasion actually is the top of the cake, so to speak, sort of a landmark of our very good relationship. Unfortunately, I met Philip only about 15 years ago, so I don't know many things about him. So I asked another Mexican, Faustino Sanchez Garduño, who actually made his DPhil in Oxford under the supervision of Professor Maini, and he was the first Mexican student, and it was very, very fortunate because the field of mathematical biology in Mexico really started then, when Faustino came back and we were forming several groups of dedicating our efforts to mathematical biology. So I'll please ask Faustino to tell you some words about who Philip is.
Okay, can you hear me? Okay. Okay, it's an honor for me to read this biographic sketch of Philip. The original version of this biographical sketch was in Spanish. I think already the Spanish version is in the homepage of the Academy. So I produced my own English version of the original one. Okay, so I am going to read it. The Center for Mathematical Biology, CMB, created in 1983, is part of the Mathematical Institute of the University of Oxford, whose founder and first director was Professor James Dixon Murray, who, among his many academic awards and distinctions, are Fellow of the Royal Society of London and an important and influential person in the world of mathematical biology. Also, he has also published hundreds of papers and supervised tens of DPhil theses. In fact, his text, Mathematical Biology, is an essential reference to all those interested in the subject. You are very welcome, Professor Murray, to Mexico. You have too many students here. Throw your book, your textbook. Since its creation, the Center CMB is a landmark to many new centers created around the world in which mathematical biology is studied and researched. Needless to say that the CMB, during its 3 decades, has also been source of a large number of researchers whose primary mathematical activity is mathematical biology. Beyond any doubt, Professor Philip Kumar Maini is one of the most prominent CMB ex-alumni. He has born in Macferfeld, it's something like Parangaricutirimícuaro. Macferf County, Londonderry, North Ireland, in the 16th of October, 1959, from Indian parents. In 1982, got his BA in mathematics at Balliol College, University of Oxford, and in 1985 got his DPhil under Professor Murray's supervision at the same university. When, in 1990, Professor Murray emigrated to the United States, Professor Maini became director of the CMB, and under his supervision, and after several years facing difficulties, donde quiera se cuecen habas, dirán en mi tierra, the CMB went through a primary transformation. Its current building is far from the 3 or 4 offices that in 1990 were part of the computing laboratory building in Keble Road. Nowadays, the CMB owns cutting-edge computing facilities as well as excellent academic atmosphere. Furthermore, there are modern and functional cubicles for graduate students, postdocs, and visiting professors. It is also worth mentioning a cafeteria and seminar rooms integrated to its infrastructure, all within the building shared by both the CMB and the Oxford Center for Industrial and Applied Mathematics, OCIAM, in Little Clarendon Street. A glance into his electronic page enlightens us about Professor Maini's diversity, creativity, and immense research production in the mathematical biology area. He has widely contributed, indeed, to the efforts tending to explain essential biological and medical processes by means of mathematical concepts and theories. Far beyond, with a nonlinear approach and working closely with experimentalists, he has put forward and studied analytically and numerically mathematical models in order to explain spatio-temporal dynamics associated to them. A partial list of the problems studied by Maini are the following: self-organization during the embryonic development, wound healing in dermis and epidermis, emergence of patterns in different biological and chemical systems, morphogenesis, in fixed domains, and in those which size and local geometry change. Cell proliferation involved in cancer tumors and aggregation of amoeba colonies, typically the Dictyostelium discoideum. The result of the performed analysis to the proposed mathematical models, in addition to its importance within the specific context of the problem from which they arose, have also been relevant from the mathematical point of view, enriching in this way the development of the science of the patterns, mathematics. His vast scientific production includes more than 300 published research papers. As co-author in a diversity of topics in mathematical biology, offers us an idea about his capacity to work and collaborate. I should add to his deep commitment as a mathematical biology teacher in Oxford, the communicating ability shown in his lectures and talks. Due to these characteristics, he has been invited as plenary speaker to many academic events around the world. His dedication as supervisor has been a constant during his profession, and the result are many DPhil and MSc theses. His ability to organize academic events is equally great, amounting to 60 workshops and conferences in whose organization he has collaborated. The editor and referee tasks regarding research projects and papers has neither been foreign to him, dedicating to them a large amount of his academic activity. For instance, he is the editor-in-chief of the Bulletin of Mathematical Biology of the Society for Mathematical Biology, member of the editorial committee or associated editor of more than a dozen of well-known journals, among them Journal of Mathematical Biology, IMA Journal on Mathematics Applied to Medicine and Biology, Journal of Nonlinear Science, Discrete and Continuous Dynamical Systems, and Journal of Theoretical Medicine. Professor Philip Maini's academic links with Mexican colleagues started in 1990, when Faustino Sanchez Garduño, now holding a tenure, a permanent position, at the Department of Mathematics, School of Science at UNAM, followed his graduate studies under his supervision, and since then maintains a close collaboration. In 1993, our colleagues Rafael Barrio and Li Mei Shang initiated joint research project with Professor Maini. As years went by, another colleagues joined the group in which Philip collaborates or has collaborated. In it are included Ramón Plaza, Pablo Padilla, Carmen Barea, and José Luis Aragón. As a host, Philip has received in the CMB to some other of our colleagues who have spent short research periods, sabbatical years, postdocs, or delivered lectures. In all instances, Philip, with his characteristic kindness, has welcomed them in a friendly and warm atmosphere. The strong academic ties between Philip and Mexican colleagues are shown in more than 20 co-written and published research papers. Professor Philip has also worked together in research projects sponsored by CONACYT and the GAPA UNAM. In his multiple visits to Mexico, Philip has taken part in several events on mathematical biology held in our country. Moreover, he is outstanding his visit within the academic exchange agreement between the Royal Society of London and the Mexican Academy of Science. Jim Murray, Philip's academic mentor, characterizes his scientific work in these terms. It's a quotation, Professor Murray. Okay. Maini is one of the major world figures in the interdisciplinary field of mathematics and the biomedical science. He is an outstanding and highly original scientist. I know most of the major international people in the world in this interdisciplinary field, and I can say without hesitation that there is a complete agreement that Maini is one of the most influential and major international people in the field. His appointment to the directorship of the prestigious Center for Mathematical Biology and the chair of mathematical biology in Oxford are only two indications of his high academic standing. The quotation ends. Okay. As a recognition of the extensive academic activity carried out by Professor Philip Kumar Maini, especially due to his valuable contribution to the development of the Mexican tradition in mathematical biology, we are very pleased to his nomination as corresponding member of the Mexican Academy of Science. Congratulations, Philip. Thank you very much for your attention.
Muchas gracias. Escucharemos las palabras del doctor Arturo Menchaca Rocha, presidente de la Academia Mexicana de Ciencias.
Buenas tardes a todos. Professor Philip Maini, distinguished incoming corresponding member of the Mexican Academy of Sciences, Dr. Manuel Torres Lavanzat, director of the Physics Institute at IFUNAM, Dr. Rafael Barrio Paredes, a member of our academy, former presidents of our academy who are present, members of the academy, students, colleagues. Professor James Murray, it's an honor to have you here. Before reading, I just want to say that while Faustino was making references to Oxford, quite a few of us, we brought nice memories of the streets of Oxford. And you may not be aware, but the people sitting right next to you, including me, we are all former Oxonians. Through the corresponding membership, the Mexican Academy of Sciences honors those foreign scientists who, besides an outstanding academic career, have contributed significantly to the development of science in Mexico. Currently, our academy counts 2,276 regular members and 92 corresponding members, among them 10 Nobel Prize winners. As we have heard from Faustino Sanchez, Professor Philip Maini is a pioneer and a world-class leader in the field of mathematical biology, as well as an important pillar in the education of students in that field at IFUNAM. He has collaborated with colleagues from this institute and the School of Sciences, and some of those scientists who have worked with him have spent extended sabbatical and postdoctoral visits at Professor Maini's laboratory since 1990. His collaboration with Mexican research groups has resulted in important publications in prestigious international journals. At present, Professor Maini is engaged in several academic activities in Mexico, including his participation in the third discussion meeting in patterning, segregation, and differentiation in complex networks, organized by this institute in order to encourage collaboration with Mexican research groups. Professor Philip Maini, it is an honor for the Mexican Academy of Sciences to welcome you as a corresponding member of the Academy. And now I proceed to give you the diploma. I would please ask everyone to stand up. I don't have a mic, but I hope everyone, this is your diploma. Welcome to the Mexican Academy of Sciences.
Muchas gracias. A continuación, escucharemos la conferencia que dictará el doctor Maini Y pedimos a los miembros del Presidium pasen a sus lugares, por favor.
Okay. So first of all, thank you very much for those very kind words that were said at the beginning there. And it's really an honor to be here and to be elected to the Mexican Academy of Sciences. So I thank you very much for all the work that colleagues have put in for that. And as was mentioned, I've been collaborating with Mexican colleagues for over 20 years now, and it's really nice to see the colleagues here. And what I've really enjoyed working with the colleagues from Mexico is not only obviously the stimulating intellectual and scientific problems we've worked on, but also that it's been enormous fun. And the warmth of the colleagues is something that I really do appreciate, and I love to come to Mexico. And my only regret is I haven't been able to come more visits. So I've really enjoyed being here. And Faustino, who was one of my first graduate students, so it was great to have him here. It's actually also nice to have my latest graduate student, Thomas, who got his doctorate on Monday, and then on Tuesday morning flew out to be here. And also lovely that Rafael is here, and Lee May and Carmen, and also it's great to see Pedro and Pablo and Clara. It's really nice to have them all here. And what also makes us very special is having Jim Murray, my supervisor, and Sheila here as well. So it actually was 30 years ago that I first was introduced to mathematical biology when, as an undergraduate student, I went to a course given by Jim, and that obviously had a great influence on what I ended up doing. So the talk today is on a tale of successes and failures. So I've never given this talk before, so hopefully it'll be okay. And I chose this because I sort of felt that when you reach a certain age, you do tend to sort of reflect on what it is that you're doing and why you're doing it. And what I decided to do was actually going to concentrate more on successes than on failures. And since I believe it's a very broad range of audience, I'm actually going to jump from different application areas, so hopefully people will find something of interest in what I'm going to describe. So first of all, there are many different modeling approaches. There are sort of statistical and bioinformatics approaches, which are what I would describe as predictive models. And the idea there is you take lots of data, for example, maybe on sequence alignment, and you try to look for sort of correspondence, say, between two genes. And if you know how one gene behaves and what it does, and if another gene looks like that, then you can say, well, maybe that gene behaves in the same way. So you're looking for that correlation. Or, for example, you might figure out from your experimental data that maybe 80% of people who have a certain genetic makeup respond very well to a certain drug. And then then you can say, well, okay, if somebody comes in and they have that genetic makeup, then we can give them this drug if they've got that illness. So that is sort of very different to the goal of what I call mechanistic models or mathematical models, which are really trying to understand the mechanism behind these things. So trying to understand, for example, what are the physical or chemical properties that, say, the gene gives to the person that makes them amenable to a certain type of drug treatment. So it's a very different approach to the same question. And of course, in some sense, these things are linked, and I think one of the big goals or challenges of the future is to link these two together. So one thing to point out is that all models are wrong by definition. And I think this quote from Turing is a really beautiful description of what a model is. So this was in 1952, and he said, in a particular model that he was developing, which I'll talk about later on, he said, this model will be a simplification and an idealization, and consequently a falsification. It is to be hoped that the features retained for discussion are those of greatest importance in the present state of knowledge. In other words, we want to abstract the key features of the problem and analyze that, and hopefully that will give us some insight. And then in another few years, the key features will change. And then we have to update our model. So this is really what modeling is all about. And then there's another thing actually from Lewis Carroll, who was a mathematician, and this is his paradox of the complete map. So the idea here is, I'll just read some of this out. There's another thing we've learned from nature, said this person, Mapmaking, map making, but we've carried it much further than you. What do you consider the largest map that would be really useful? About 6 inches to the mile. Only 6 inches, exclaimed Meinherr. We very soon got to 6 yards to the mile. Then we tried 100 yards to the mile, and then came the grandest idea of all. We actually made a map of the country on the scale of a mile to the mile. Have you used it much? I inquired. It has never been spread out yet. The farmers objected. They said it would cover the whole country and shut out the sunlight. So we now use the country itself as its own map. And I assure you, it does nearly as well. Okay? Now, the reason why I put this up is because of a fundamental misunderstanding of mathematical modeling in the biological community. Because the biological community thinks this is what we're trying to do. And therefore, quite rightly say, you can't do that. And we can't do it because we don't know the details of the country to this level. But we're not trying to do this. We're trying to do what Turing said. So I'll give you one example of that. Oh, the other thing is, this other quote, all models are wrong, as I mentioned, but some are useful, from George Box. So I'll now give my first example, and this is actually work that we've been doing. So this is the mouse embryo, and it has these cells here called the AVE cells, a group of cells, which migrate up the surface in early development. And it's very important they do that because they orient the anterior-posterior axes of the embryo. And if something goes wrong with this, it really totally destroys the development of the embryo. So we have a colleague, biological colleague, Shankar Srinivas. So he came to us and he said, could mathematical modeling explain why these cells move up? And we said, no. Maybe there is some chemical here that's pulling them up. Maybe there is some force at the bottom pushing them up. We don't know enough of the biology to answer this question, so sorry, we can't. So then he said, well, actually, another thing I wanted to point out, he said, was I've discovered this very strange behavior. During the movement of the cells up, I noticed this sort of structure, which is called a rosette. So he was the first person to discover the formation of rosettes in the mouse embryo. They'd been discovered a couple of years earlier in the Drosophila embryo. And so he said, can you explain how this forms? And we said, no. There's not enough known about the biology for us to really describe this. Now, luckily, he is a very patient man. So then he said, could you use a mathematical model to understand why rosettes are necessary? And then we said, well, yeah, that's a question we might be able to answer. So along with his research group and with graduate student Aaron Smith and colleagues David Kay and Ruth Baker, we started to look at this problem. So here's just some more of these. You can see these rosettes here, and in time how they change. And what we did was we used a vertex model, which people like George Oster and Mike Wellicke sort of introduced over 20 years ago, and Honda and very many people have looked at this. And basically the idea of the vertex model is that each one of these is a cell, and you essentially solve Newton's laws of motion for each vertex, move the vertex, and then redraw the system. And what we did then was that we made such situations that cell would divide when they reach a certain size. And what we did was then we introduced these cells here, which are green, and we said, let's assume that there's some mechanism that's moving these cells up. And let's assume that rosettes can form. So when a number of cells come together, we let them stick together and form rosettes. This is not a particularly interesting video, actually. Movie, it'll just show these things moving up. And you see they move up in a very coherent fashion. And then they keep moving up, and they move up to halfway up here, and then they stop, which is the biological observation. Okay, and that's what you would expect. Of course, you put a force to say things should move up, then of course they'll move up. But then we started to compare with the data. Now, we can't make total comparisons with numbers, but we can make comparisons with relative numbers. So for example, one of the students that worked with Shankar, her job was to count all these cells and count how many sides each cell had. And therefore, what are the number of polygons, average polygon number, and how do they vary in different parts of the embryo? So for instance, here you notice that early you get this number, and late you get this number. And then we can compare with the simulations. And what you can see here is that it captures the decrease in the average polygon number. Then we can do things, actually by polygon numbers. So for instance, here that means there's the 5-sided polygon, 6-sided polygon, et cetera, et cetera. And what we can do here is by simulating the model at different time points during the course of the simulation. So this is not a very good color scheme, actually. This is the simulation. This is the actual results. What we end up getting is that qualitatively we get the correct level of frequencies. So in other words, we see here that the 5-sided polygons, there tend to be a lot of them, quite a few 6-sided polygons, and then not so many of these other type of polygons. And then we can follow this over time, and we get the same amount. So what's very surprising is that this very simple model, which actually has very little biology in it, is qualitatively similar to what's observed experimentally. So now we said, let us suppose now we don't allow rosettes to form. What will happen then? So we run the simulation again, and now this time rosettes are not allowed to form. What you can see is this breaks apart. So the prediction of our model was, if you have a mutant in which rosettes don't form, you will not get a coordinated movement of the cells up the embryo. And so Shankar looked at the mutants, and that's precisely what he found. So then we were very, very excited about this. Oh, that's just in case the videos didn't work. That's in there. So the model prediction for mutants which do not form rosettes, migration will be disordered, and sure enough, when he looked at the mutants, this is what he found. So then we submitted this paper to PLOS Computation, to PLOS Biology. We're very excited with this result, and the paper was rejected. And the reason why it was rejected was one referee said, you were trying to model the embryo of the mouse. You can't model the embryo of the mouse because we don't have sufficient data to model it. In other words, that referee was wanting the one-to-one map. So for example, that referee was saying that when I'm flying from London to Mexico City, the little map that comes on the screen is useless. It's only that size. What I actually need is a map that's 8,000 miles long. And if I don't have that, I shouldn't bother with any map. That's essentially what they're saying. And we said, we agree with what you say, but that's not what we're trying to do. So luckily enough, there was a good editor, and the editor then disqualified this person from the next round of the refereeing, and then this paper was actually published yesterday. But it gives you a sense of the fundamental misunderstanding between what biologists think a model should do and what mathematicians think a model should do, or a model can do. Now, where modeling has been very, very successful has been in ecology and epidemiology. So now shifting from developmental biology to ecology. And in fact, in the UK, models are used quite a bit to inform the policy. Because in the UK, we seem to get all these diseases of animals all the time. And then policy modelers work with the government to decide on the policies. One of the best known models, so this is a problem that happened in the UK in the early part of the last century. And this is work, actually, one of the first things I did as a postdoc with Jim Murray, that around about 1900s, somebody had the very bright idea of bringing the North American squirrel to the UK and released it, and they have now driven out the indigenous red squirrel. And the grey squirrels are a pest. So there's there's a red squirrel. So that's the native of the UK. Very, very nice. And then along came this guy, okay? Bigger and tougher and better, and outcompeted the small guy. This is, I apologize for this. This is a sort of, if you think of the UK as being a triangle, then this is the sort of corner of the triangle, and they were introduced around here, and these black spots are where red squirrels were seen, and the sort of empty spots are where grey squirrels were seen. And now you see, as we move on, this is 1964, this is 1979, you can see that there's virtually no red squirrels. It's all grey squirrels. So when one does, oh yeah, and then this is very good here, and I really wish they would still write scientific papers like this. This is a scientific paper, which it quotes some guy saying that that the grey squirrels attacked one of his trees and ate all the plums on the tree. This club offered 6 pence, which is about 3 American cents, for, presumably by meaning a head, they didn't literally mean if you brought in the head of a grey squirrel, they probably meant you brought in a dead grey squirrel. This is actually quite a lot of money because this is a way back in the early part of the last century. But no matter how many squirrels they killed, they weren't able to get rid of them. So why is that? Can we understand that with a mathematical model? And the answer is yes. So this is using the model, Lavolterra model, from a very long time ago. And this is work with Akira Okubo and Mark Williamson and Jim from quite a long time ago. And basically what we find, I'm not going to go through the details of this equations, These are just gray squirrels and red squirrels. See, I use color coding here. And this is the interaction terms, competition terms. And what you actually find is there's a bifurcation. So in one level of the bifurcation, you get this behavior. This is a phase plane, and what this says is that no matter where you start, you always go to this point here. So the gray squirrels always win, and they exclude the red squirrels. So if you're in this part of parameter space, it means that if the gray squirrels are high and you kill a lot of them, you still go back. There's nothing you can do to prevent going back to there. If you're in this part of the domain, you can either go to there and have all gray squirrels, or you can go to there and have all red squirrels. So in this situation here, if you killed a lot of the gray squirrels, you would get rid of them. In this situation, you would never be able to do so. The difference between the two is in these interaction parameters. So what this is telling us, in fact, in the case of the gray squirrel, is killing the gray squirrels will not get rid of them. What you need to do is change the environment by changing these interaction terms. Now then the question asked is that a useful result to find out? Well, in one sense it's useful because it's saying what you're doing will not work. But in another sense it's not useful because how do you change the environment? So can you really use that model? Oh, and then I just show this video here that the student called the Squirrel Wars. And so this is, introduce some gray squirrels here, here, and here into a population of red squirrels. And what you end up getting is an invasion of traveling waves as the gray squirrels, according to that model, will invade. And so you get all gray squirrels and all the red squirrels disappear. Okay? But like I say, the problem with that model and that application was that you can't change the environment. So now let's move forward some 70 years to medicine. So Bob Gatenby, who is a clinician, he's a cancer radiologist, he looked at cancer in a way that people hadn't looked at it before. He looked at it in an ecological setting. And he asked the question, if you think of cancer as a pest, how can a pest overcome, if you want to move into another population, how can you overcome that population? Well, there's only two things you can do. Either you kill the other population, or you breed faster than the other population. Now, it's known that the pH of the body favors normal cell division. It's also known in something called the Warburg effect that tumor cells undergo something called the glycolytic pathway to generate energy, and this is 20 times less efficient than the normal cells. So the question is, you've got an environment that favors the normal cells, you've got an invader that's producing energy an order of magnitude less efficient than the host, and the invader wins. How did that happen? Well, the hypothesis here is that it changes the environment, because a byproduct of the glycolytic pathway is lactic acid. Lactic acid is poisonous to normal cells, and also it changes the pH to where the tumor cells can divide quicker. So in other words, in the Lavolterra model, it changes the parameter values. So they wrote down a Lavolterra type model. I won't go into all the details. This essentially is a Lavolterra model. And what they found was this behavior here. So everything has been normalized to 1. So what they did here was to say, we start off with all normal cells. The normal cells are these dotted dashed lines here. And then we put some tumor cells in, and we see what happens. And just like in the squirrel wars, we get a traveling wave. So here's the tumor cells. Here's the normal cells, and here is the lactic acid. And this propagates, just like that movie I showed. This propagates along. On the other side of the parameter space, you get this behavior. Here's the tumor cells, here's the normal cells, and here's the acid. What you can see here is there's a gap. So that's something different about this model into the Lavolterra model. There's a prediction of this gap. So then he is experimentalist, so he went to look for the gap. And there's the gap there. Now, it's important to point out that this is a very controversial, still controversial, because some people argue that the biological process you have to use to get this picture can influence and form this gap. The fixing can form the gap. And if you do this without fixing, you still get a gap, but it's not as impressive as that. So it's still controversial. But one of the obvious outcomes of this is a suggestion for therapy. Oh, before I go on to that, these are also then what they did, the pHs. So this is the tumor here and the normals here. And these are taking cross-sections at different cross-sections. And so that's pH. So that means the gradient of acid looks like this, qualitatively the same as they suggested. So they decided, well, if the tumor is using acid to invade, if we neutralize the acid, the tumor shouldn't invade. So that's what they did here. So they take the poor mouse and they chronically give it bicarbonate. So the drinking water, anything the mouse drinks has got bicarbonate in it. And the result of that is it changes the pH of the mouse. Then they put into the mouse tumor cells, which they label with GFP, and these tumor cells metastasize to the lungs. And they use one set of just normal mice, and you can see here, this is in the lungs, you see there's lots of green here, which are the metastases, and these are the treated mice, and you see there's lots of black here. There's no metastases. And here, when you do the data analysis, you see in the untreated, there's a lot of invasion. In the treated one, there's very little invasion. So that's consistent with the model and was motivated entirely by their modeling work. Of course, we don't really worry too much about whether we can cure mice of cancer. We want to figure out, can we cure humans of cancer? And so what we've been doing recently, in fact, this paper was accepted last week in the British Journal of Cancer, and this was Natasha Martin, who was a graduate student of mine, and Eamon Gaffney, who's a colleague in Oxford. And then these are the experimentalists in Roby, Bob Gillies, and Bob Gatenby. And what we asked was, now let's do this modeling in humans. And when we do the same model in humans, because humans are different to mice, they have different parameter values, what we find is the prediction that the acid buffering will not be as effective in humans. But now, because we've got a mathematical model, we can explore other parameters. And one of the predictions we make of the model is, in fact, elderly people that have renal failure which corresponds to one parameter in the model being different to everything else, they would respond to the bicarbonate treatment more in a very effective way. We can also say what would happen if you added other buffers, and we can figure out what the properties of these buffers should be. We can get a pK value, which is linked to the dissociation constant, of what it should be for the ideal buffer. Actually, I should point out, when we first realized that this was limited, we said to Bob, why not give more and more bicarbonate? But he pointed out bicarbonate was toxic. So you'd kill the person if you gave a lot more. So this model has been validated, at least for the mice in here, validated by experiments. And what we're now hoping to do is to start looking at these combination treatments that are predicted by the model to see if they will help. So the model is suggesting possible combination treatments that might work. So staying with physiology, this is perhaps the most famous example, the Hodgkin-Huxley model of 1952, which is the squid giant axon, not the giant squid axon. Okay? And they looked, so here's a nerve cell neuron, and there are going to be some talks tomorrow on neurons. And the idea here is to try and understand how electrical activity propagates through the neurons, and in this way the neurons communicate with each other. So when they were looking at this, they basically get this action potential. So this is the voltage that sort of propagates along, and that's what carries information from around the body. They wanted to understand this, and they postulated that, well, the potential was related to a number of currents. These currents were due to the motion of sodium, potassium, and other ions. And they postulated that these ions moved through channels. And they said these channels had gates. And the gates opened or closed depending on the voltage. So they had other equations then for these I's. So it had quite a complicated nonlinear system of ODEs. And when they solved these and fitted it to the data, they determined what the nature of the channels should be in terms of how many channels should be stimulated by, how many gates should be stimulated by sodium or potassium, et cetera, before the biological technology was able to probe at that level. And then when the biology technology was able to probe at that level, they found that the model predictions were spot on. And so that happens, you get one of these. Nobel Prize. And in fact, this model is used really in whole heart models in a lot of, it's been a building block for a huge amount of work in electrophysiology in all sorts of areas. So I'm not going to talk about glioblastoma, which is another success, because I think that Jim is going to talk about that. And another success is marriage, which I'm not going to talk about because Jim's going to talk about that. And then I thought, what's common about these two things? The only thing I can think of is they both give you headaches. But that's for the talk later on. And now we come back to Turing. So Alan Turing, whose 100th anniversary of his birth is to this year, in the summer. And he came up with a model for biological pattern formation in 1952, where he said that he wrote down these sets of equations. So that's just in case there's some mathematicians in the audience. what he said was he was wanting to understand, for example, if you've got a tree or a branch that's growing, if you look down at the top, you would see a circle. You see symmetry. And then something branches off it. So symmetry is broken. And those of you who are physicists in the audience will know that that's really what physicists always think about. How does symmetry break? Very exciting thing. And he came up with the idea of diffusion caused the symmetry to break. And diffusion was a destabilizing element. So that's totally counterintuitive. And he proposed the idea that you had chemicals which he called morphogens. Due to diffusion, the symmetry in the chemicals were broken, and that led to the symmetry breaking of the structure. So for example, if one of these chemicals was a growth hormone, and the growth hormone was symmetric, then the tree would grow symmetrically. And then you got the symmetry break in the growth hormone, which means there was more growth hormone in one side, you got the bud. So that was his idea of biological pattern formation, and it was based on this patterning principle: short-range activation, long-range inhibition. And now this model is hugely controversial. So one of the failures of this model was in trying to explain, this is the, but then unfortunately some nasty biologist came along, did some experiments and showed that each one of these was formed independently of the others. In other words, you could remove one of these and keep the others, totally contradicting the model of Turing. And in fact, what it turns out that's happening here, actually this one here takes a long time to go to the next slide. Sorry about the, you probably can't see this, which is probably a good thing because it is a complete mess. You've got all these chemicals, all these proteins, I should say, interacting with each other. And you have this huge model system that you need to solve. And what happens is you get a cascade of reactions occurring. So you get a gradient. That gradient stimulates another gradient. Those two gradients stimulate another pattern. That stimulates another pattern, and in that way you develop these 7 stripes. Now, this is work by von Dassau and Gary Odell, quite a long time ago now. And they modeled this, and of course, when you start modeling something like this, you don't know what the parameter values are. So they literally did millions of simulations, and they couldn't get the right answer because you get there's a lot of work on mutations, so you've got to check your model against these pair rule genes and against mutations. They couldn't get it to work at all. And then biologists had postulated that there were 2 other links between here. And when they put those links in, they found that it didn't matter what parameters they used, they always got the right answer. So they used the model to verify and, in some sense, validate the idea that there were these connections. And then, in fact, Hans Othmer and Rika Albert later on modeled this as a Boolean network. And what they showed in their model was that it was the topology of this that gave the pattern, not the strength of the parameter values. So to spend time and money measuring the parameter values was a total waste. All you needed to know was the network topology. Now, the model gives lots of spectacular patterns. So this is work from Hans Meinhardt, and this is a seashell, and this is the reaction diffusion model of the seashell. This is another one, this is the model, another one, this is the model, and this is a fish, and model of the fish, another fish, model of the fish. This looks very spectacular. of course, just because you can reproduce the pattern doesn't mean it's the right thing. And in fact, there's a lot of controversy about this because in recent work, building on work from a long time ago, Bart Ermentrout and George Oster and colleagues, they suggested that there's not a morphogen involved in this system, it's actually neural interactions. And in fact, when you write down the neural interaction model, it's identical to the Turing model. So from a mathematical point of view, you can't distinguish between the two. But they were able to do experiments to really give a very, very good case that in fact it's a neural model rather than a morphogen-driven model. Now, the Turing model has been verified in chemistry. This is something called the SEMA reaction, and you get these patterns precisely as predicted by the model. And that's just the model predictions of those things. And then here, so this is that fish that's here, but this is earlier version of that fish. And what you find here, this is work by Kondo and Asai, that these fish have stripes, and as the fish grows, stripes pop up in the middle to preserve the wavelength. It's precisely what's predicted by the mathematical model. Another way stripes can rise is from something like, this is the simulation. It's easier to see the simulation than see the actual thing. And this propagates along, and it splits this way. And you can see this happens in the real thing. This is also work by Kondo and Asai. And this is one thing which is pretty amazing. This, again, is Kondo's work. So they took a zebrafish, and they ablated the stripes here, and they asked the question, what happens now? How does it regenerate? Now, I would have thought the stripes will come back again, but this is what happens. Precisely as predicted by the mathematical model, says that that should happen. And then they've also done work where they've, by taking the leopard fish and by doing mutations, they've generated different types of structures. And then they've taken their model, and always people say, well, you've got these models, you've got lots of parameter values. You can produce anything with lots of parameter values. What they did was they varied one parameter. And as they varied one parameter, they got all these different patterns. So if you wanted to validate this model, you would need to find the morphogens, the chemicals that are interacting. And there's a lot of work, now experimental work, in trying to find what these chemicals are. So for example, in the mouse, it's thought that Wnt and DKK are involved in determining the hair follicle spacing. In limb development, it's thought that BMP, bone morphogenic protein, fibroblastic growth factor, may be involved. And in vascular calcification, it's thought BMP2 and MGP may be Turing-type morphogens. It's still controversial. So it turns out that all of these interact qualitatively in the way they need to interact to give diffusion-driven instability. But the question is that what's actually happening? Now, within the fish, they haven't been able to find the morphogens. And in fact, Kondo's recent view is that in the fish, maybe there aren't any morphogens. But maybe the diffusion term comes from the cells being able to contact non-local neighbors. So the question now you would ask is this a success of the model or is it a failure? Well, it's a failure of the model because the model said there should be morphogens, and they look for morphogens, they can't find it. Can't find them. But you could also say it's a success of the model, because the model made a prediction that there has to be a non-local interaction that's of a diffusive nature. And when you take their model and you put in this long-range for non-local interactions of the cells, you get a diffusive part. So mathematically, you get the same model. And in fact, Kondo was driven to look for this non-local interaction when he couldn't find the morphogens. He was driven by the model. So the model is wrong, but all models are wrong, but some are useful. And so the last thing I'll talk about, we're all getting hungry, and this is slime mold. And these are bacteria that when they're running out of food, they form aggregation patterns. And this is part of the life cycle. So they create these chemical waves, and they aggregate in response to the chemical waves. Now this was modeled quite extensively in the 1990s by lots of different people. I'm not going into the detail. This is just the mathematical model that gives rise to that. That's not really important. What I want to get is the verification of the model. So basically what the model says is the cells move up the gradient of the chemical. And that combined with when the cell receives a chemical signal, it emits the chemical signal. If you put those two things together, you get all these patterns. And people have done experiments. So one experiment they did, I don't know why anybody thought of this, but if you treat the slime mold with caffeine, now I don't know why they thought of putting caffeine in, they get, instead of getting a nice aggregation, they get an aggregation with a hole in the middle. And now they've figured out what the caffeine does. So you can go into the mathematical models and you can change the parameter that the caffeine changes. And if you do that, you get that. Okay? Now, then you present that to the biologist, and the biologist said, that's totally useless. I know that that's what happens. You haven't told me anything new. So you're just wasting your time. Right? If I can do the experiment and get the result, and you come 2 years later with a mathematical model that gets the result, what use is that to me? Now, when they measured these waves to which the cells respond, what they find is that if you look at the wave speed against wave number, it looks like this. It decreases. So the biologist said, well, the wave propagation is due to the biochemistry and the physics of the system. So if the wave propagation is changing over time, it means there must be biochemical changes occurring in the system over time. Because they said, if all the parameters were constant, this is what you would get. So this is work we did with Thomas Hofer, who was a graduate student at the time, and Jonathan Sherritt. We solve the model, we keep all the parameters constant, and this is what we get. In other words, the verbal reasoning that to get this, you must change the parameters is wrong. The system self-organizes and automatically changes the parameters. Now that's something, an insight that you would not get without a mathematical model. And this mathematical model takes into account just 2 or 3 of the processes that are going on. And what it suggests is to look for changes in the biochemistry is to actually maybe waste your time going along here. So to summarize, I think, you know, are mathematical models useful? Are they not? I presented some examples. You can decide what you think. Maybe we should have a vote at the end, like in the game show. One thing a mathematical model does is it makes you write down assumptions in a very concise way, in a way that verbal models don't. There's a very spectacular example of this, which is in the process of the formation of cancer. A cancer, when it starts to grow, it causes more vasculature to form. It's a process called angiogenesis. And it was thought that if you gave anti-angiogenesis treatment to stop that from happening, you would cure the cancer. And this hasn't happened. Now, if you write down concisely what are the assumptions of that verbal model, the assumptions are that delivery of nutrient to the tumor is a monotonic increasing function of vascular density. And therefore, if you reduce the vascular density, you reduce the delivery of the nutrient. Now, millions and billions of pounds and dollars have been spent on anti-angiogenesis treatment. Can anybody guess how much money has been spent on trying to verify that hypothesis on which the model is built? The answer is 0, because people didn't realize that that was their hypothesis. And in fact, the data that we have shows that that hypothesis is totally wrong. And that's why this doesn't work. Writing down a mathematical model, you would have immediately seen that that was the first thing you needed to check before you went to spend billions of dollars. And the last thing to say is, of course, you really must talk with experimentalists and really link in with them. And even though it's very frustrating, and even though biologists are very annoying people, you still have to talk to them, and I'm sure they would say exactly the same thing about me. So I'd like to thank you for your attention, and thank you again for organizing this, and for all my colleagues for arranging this, and I've really enjoyed it, and thank you for your attention.
Thank you, Philip, for your exciting lecture. Are there any questions? To ask questions.
Well, maybe I can elaborate a question that I like very much your talk, and especially the ideas that you bring up. Philosophy, so to speak, of modeling and what it implies in science and so on. And my question or a comment that maybe you can comment to my comment is, or is more a question. Sometimes when you interpret the data or in an experiment, you interpret data in terms of a model that you have in your head.
Yeah.
That means that there is a feedback between modeling and experiments and back. And maybe you can comment or elaborate on this?
That's actually extremely important, because I think that's one of the key things that's wrong with science today, because sometimes you ask, like for instance, a classical example of this is the growth of spheroids in tumor growth. So we've tried to grow spheroids because there's a very nice model about the spheroids. Spheroids grow, they reach a certain size, and then you get a necrotic core because the oxygen can't reach the center. Now, when we try to grow spheroids, we find that sometimes you get spheroids, sometimes you get all sorts of funny-looking things. And then you talk to the experimentalists, and they say, oh, yeah, But those ones, the experiment didn't work. So we got rid of them. So it's self-fulfilling. If you go in saying, this is what I think is happening, and if my experimental data do not agree with that, it meant the experiment didn't work. And so you ignore that. We have a very nice example of that with work with Paul Klesse, who is from the STARS Institute, and this is work on modeling movement of the neural crest cells in early development. And we had a meeting with them because what he can do, he's a fantastic experimentalist, he can do things like he can sort of chop different parts of the domain and move them back and forth and all this and that and other thing. So we had a meeting with him and we said, right, we're going to do 4 experiments. Now you go back to Kansas and you do these 4 experiments. Louise, who was a graduate student, she'll do the experiments in the model. We won't talk to each other. Then she will go to Kansas and we will open the results and see what the results were. 3 of the results agreed perfectly. And the 4th one, the model was wrong, because Paul had shown that when you did a certain experiment, you got a nice invasion, and the model said you wouldn't get any invasion. So Louise, being a meek graduate student, said, I'm very sorry, the model is wrong. And then Paul said, actually, we did 50 experiments. 5 of them are the data we present with the cells moving along. The other 45 didn't work because the cells didn't move. Precisely as predicted by the mathematical model. But his model in the mind was the cells should move. Now it makes you ask the question, how much of that is already in the literature that we don't know about? So it's a very, very good comment you make.
So you're advising us not to read the literature or something? No. Most of it is wrong.
The point is that this is a very crucial aspect of science in general.
It's very crucial.
And it doesn't often discussed, as should be,
that's
in
right.
my opinion.
That's absolutely.
So that's why I wanted your...
I couldn't agree more with you. I mean, I think that's a really serious problem. I absolutely agree with you. Yeah, that's right.
Well, I really liked your talk, very interesting one, but I will try to make you a question as an annoying biologist. In the sense that in biology, you say, well, people say that you cannot say that you understand something unless that you incorporate evolutionary ideas. So the concept is that all these stripes of the fishes or the spots in animals or whatever, they are the result of adaptive and evolutionary process, which they have like the effect of avoiding predators or finding a mate. So, and I don't see anything of that in the equations, and so I would like to say, why didn't you incorporate that?
Yeah, this is another very good question, and the idea, I mean, there are people working that in what's called sort of Evo-Devo, they call it, and so Turing totally decoupled the two things from happening. So in what Turing was trying to do is he was trying to say, what are the mechanisms by which you get a certain pattern? But so you write down these 2 equations, and you've got parameters in there. That's where the genetics is encoded, because these parameters are production rates, death rates, diffusion coefficients, et cetera, this is where those things are encoded. So if you wanted to view it in that context, you would then say that, well, the parameters were selected so that, but we don't know how they were selected, but the parameters were selected to be in that parameter space. And then the model says, if you're in that parameter space, this is the mechanism that gives rise to the patterns that you observe. Now, what you would really want to do is to start off with the system and have some sort of evolution where you would sort of simulate the system and you would maybe give points to certain types of pattern. And then you would say, okay, if these ones win, then when they genetically go to the next thing, then you see whether they can evolve. But that's something that, in fact, some of the bioinformatics people in Oxford want to do with us, but we haven't got around to doing it. And then that's where, because that would be very interesting then to see whether you saw the different pathways in evolution. In fact, that's something that Jim has done some work on that by in looking at limb development, and you can see that by changing parameters, you get various sort of prehistoric patterns of limbs.
We did also the same in sea urchins.
Yeah, that's right. With the
which has no biological advantage. It's just a parameter that evolves within a bifurcation tree, and you reproduce the shape of the sea urchins in different evolutionary times. Since you mentioned evolution, which is the biology part of the thing, and Philip mentioned that it's 100 years of Turing. Yesterday was 200 years of Darwin. It's a 200-year anniversary of Darwin. So if we didn't dedicate this meeting to Philip, we should dedicate it to Darwin. Yes, and Turing. But I mean, since we cannot do that, I think one person that encompasses both is Philip. Yes, this is a mathematician and a biologist. Another question?
Thomas.
I could ask what happens to Turing when you add delays in, but I think that's a bit too specific. I knew, yes, I know. But my question is simply, when do you stop adding to a model and you say, that's it, either this is going to work or it's the wrong model?
Yeah, that's actually a very good point, because, you know, at what point does the model start being like the one-to-one map of the Earth? And I think that then that sort of backtracks as what do you want to use the model for? If you want to use the model as predictive, then I think you do need the one-to-one map, because then the idea would be that if you want to figure out what happens if I change a certain parameter, if you actually had a faithful reproduction of the cell on your computer, you wouldn't have to do the experiment on the cell, you could do it on the computer. So in that sense, the answer would be, yeah, you have to keep on and on adding things. But if your view of the modeling is, I want to try and do the abstraction and get some idea of some maybe principles that might be at work, then I think as soon as you start adding more and more, you lose the detail of all of that. And there is a move to develop really, really complicated big models. Now, the models I presented are all either one equation or two equations. And I didn't present any models that are hundreds and hundreds of equations, because I think that they don't give as much insight to what's really happening. They're answering a different question. But I guess there's the other point, which is, you know, maybe you just totally got the wrong track. And if you over-parameterize the model, it eventually will agree with what you see, because you've got so many degrees of freedom. So that's a good, very good question. Yeah.
Any more questions?
Hi. I must say I was working with Philip 20 years ago and working in mathematical biology, but now I'm most experimental biologist. So my question is, the model you have presented here, it's most of them has a very small amount of variables, you know. So you presented the neuron model of Hodgkin-Huxley in the. This is a very very limited. Yes, but how can we make a successful mathematical model in a bigger time scale and space? No, because we see for instance in the brain, you know, in the very small region, you have a lot of detailed different cell types. For example, in hippocampus, you have two type of principal neurons and more than 25 type of interneurons, and they are distinct in their morphology and in their chemical inside. So when you have a large amount of variables, how can this mathematical model make some successful prediction?
So I'd give sort of two answers to that. So one answer would be that maybe mathematical modeling can't in that situation. Maybe you just have to do experiments. You know, for example, if I want to measure temperature and I use a microscope, I won't be able to measure the temperature, but that then should I never use a microscope? It's useless, can't measure temperature. I've used it for the wrong thing. So maybe this is a situation where the mathematical modeling cannot be used. Another aspect of that would be that maybe at the moment we've got the wrong mathematics. That maybe if we had some other type of mathematics, it would allow us to deal with the complexity. And therefore one could write down a very complicated model, which is, you know, you could write down a model of what you've described. It would be really, really complicated. The mathematics we have at the moment will not allow us to have insight from that system of equations. Maybe there's another mathematics out there yet to be discovered that will make it understandable. You know, I don't know.
Yes. It's a very small question. Just you know, when you have a lot of details in biology, you know, one of the big complaints of biologists to the mathematicians, because the mathematician says this details is ignorable. No. You know, the big complaint is many details. Maybe we believe it's essential, but we still don't know why or in which way. But mathematician came and say this is not an important detail. So how can one make the distinction which details you should use for your modeling?
Well, it depends on what question you're trying to answer. So for example, in that model of the dictyostelium that I mentioned, in order to answer the question of what happens about how the wavelength changes, what we did in that model was simply have a small set of equations to describe phenomenologically the production of the chemical and the chemoattraction. But then when you look at the model of what happens when you add the caffeine affects the detail of the intracellular pathways that are going on inside the cell. So it means the model is inappropriate to describe that. What you need to do is look at the intracellular behavior, see how the model affects the behavior of that, and then translate that, upscale it to the other model. So this is another thing where there's a misunderstanding of there's the model for something. There isn't. I would say there's a model for the question you're trying to answer. And if you're modeling the same thing but trying to answer a different question, you probably need a different model with different levels. And that's why I think the mathematician is wrong when they said these details are not important. They may be not important for the question the mathematician wants to answer. They may be very important for the question you want to answer. But mathematicians are very arrogant people.
Okay. Okay.
I finished. Yes. Just going back to the point of the final talk of the final part of your talk. I think you are telling us something about the reflection of the modelization, mathematical modelization into biology. in some of your examples, the space and time scales are certain, maybe microscopic, let's say, and maybe the time is short time because it's not evolutionary things explicitly incorporated into the models. So this is one point. This is you have talked mainly in microscopic level. When you go up to the bigger space level, so you must, well, from the modelization point of view, you must incorporate which kind of interaction at microscopic level you have in order to produce this microscopical behavior or patterning or whatever. So from modelization point of view, the modeler should take into account these changes in the scales, in the space and time as well. In this change, going from the microscopic to macroscopic, maybe the mathematical models at certain level is not necessarily useful to when you change the scale. So but this is the point. The model must know that. I finished.
You know, no, absolutely, that's right. No, I agree with that, yeah. I mean, these models are considering the cells as a black box. So it's modeling from the cell level upwards. But you're right, I mean, all the parameter values, growth rates, death rates, etc., are determined from coming at a microscopic scale. And ideally, you want to link all of those together. Yeah.
Thank you very much for your attention.
Thank you.